Mitosis and meiosis prelab. Brief description. Prior to mitosis, the chromosomes in the nucleus are replicated. The nucleus then divides. Nuclear division is usually followed by division of the cytoplasm. In mitosis, there is one such division. Mitosis is division in development of an adult organism from a single fertilized egg, in growth and repair of tissues, in regeneration of body parts, and in asexual reproduction. In mitosis, the parent cell produces two daughter cells that are genetically identical. The term daughter cell is conventional but does not indicate the sex of the offspring cell. Mitosis can occur in both diploid and haploid cells. Interphase. Most of the cell cycle is spent in interphase. During interphase, the cell is growing and metabolic activity is very high. Toward the end of interphase, new DNA is synthesized and the chromosomes are replicated. Each chromosome of homologous pair is then composed of two sister chromatids. Prophase. In prophase, the chromosomes appear condensed and the nuclear envelope is not apparent. Metaphase. In metaphase, thick coiled chromosomes, each with two chromatids, are lined up on the metaphase plate. Anaphase. In anaphase, the chromatids of each chromosome have separated and are moving toward the poles. Telophase. In telophase, the chromosomes are at the poles and are becoming more diffuse. The nuclear envelope is reforming and the cytoplasm may be dividing. Cytokinesis. In cytokinesis, there is division into two daughter cells is completed. Mitosis versus meiosis. In diploid organisms, the genome is composed of homologous chromosomes. One chromosome of each homologous pair comes from the mother, called a maternal chromosome, and one comes from the father, called a paternal chromosome. Homologous chromosomes are similar but not identical. Each carries the same genes in the same order, but the alleles for each trait may not be the same. In garden peas, for example, the gene for pod color on the maternal chromosome might be the yellow allele, and the gene on the homologous paternal chromosome might be the green allele. Mitosis and meiosis are not completely similar. The cell cycle is something like a dance routine or football play. It is a process that runs continuously but is impossible to stop the action at any point for analysis. From a dead collection of cells on a lab slide, it may be hard for you to grasp the dynamics of mitosis. Here is a movie clip of mitosis in a plant cell. As you view the clip, try to identify each stage of cell division. Lab procedure. Number 1. Determine the approximate duration of the entire cycle for the cells you are studying. Number 2. Looking at the slide, count and record the number of cells in the field of view that are in each phase. Number 3. Determine the total number of cells counted. Number 4. Determine the percent of cells that are in each phase. Number 5. To calculate the time in minutes for each phase, multiply the percent of cells in that phase by the number of minutes for the whole cycle. In this lab, you will count at least 200 cells by moving your slide so that you view several fields.